Dear students, today we are going to discuss about the topic metabolism of amino acids. Proteins are the most abundant organic compounds and constitute a major part of the body dry weight, that is 10 to 12 kg in adults. They perform a wide variety of static and dynamic functions. About half of the body protein is present in the supportive tissue, while the other half is intracellular. Proteins are nitrogen containing macromolecules consisting of L-alpha amino acids as the repeating units. Of the 20 amino acids found in proteins, half can be synthesized by the body and called as non-essential amino acids, while the rest have to be provided in the diet called essential amino acids. The proteins on degradation release individual amino acids. Amino acids are not just the structural components of proteins. Each one of the 20 naturally occurring amino acids undergoes its own metabolism and performs specific functions. Some of the amino acids can also serve as precursors for the synthesis of many biologically important compounds, example melanin, serotonin, creatine, etc. Certain amino acids may directly act as neurotransmitters, example we can say glycine, aspartate, glutamate. Protein metabolism is more appropriately learnt as metabolism of amino acids. An adult has about 100 gram of free amino acids, which represent the amino acid pool of the body. The amino acid pool may be an oversimplification of the facts, since there is no single compartment, rather several compartments exist. Glutamate and glutamine together constitute about 50% and essential amino acids about 10% of the body pool. The concentration of intracellular amino acids is always higher than the extracellular amino acids. Amino acids enter the cells against a concentration gradient by active transport. The amino acid pool of the body is maintained by the sources that contribute and the metabolic pathways that utilize the amino acids. What are the sources of amino acid pool? Turnover of body protein, intake of dietary protein, and synthesis of non-essential amino acids contribute to the body amino acid pool. What is protein turnover? The protein present in the body is in a dynamic state. It is estimated that about 300 to 400 gram of protein per day is constantly degraded and synthesized, which represents body protein turnover. There is a wide variation in the turnover of individual proteins. For example, the plasma proteins and digestive enzymes are rapidly degraded, their half-lives being in hours or days. The structural proteins have long half-lives, often in months and years. How this protein turnover is controlled? The turnover of the protein is influenced by many factors. A small polypeptide called ubiquitin tags with the proteins and facilitates degradation. Certain proteins with amino acid sequence proline, glutamine, serine and threonine are rapidly degraded. There is a regular loss of nitrogen from the body due to degradation of amino acids. In healthy adults, it is estimated that about 30 to 50 gram of protein is lost every day from the body. This amount of protein must therefore be supplied daily in the diet to maintain nitrogen balance. The purpose of dietary protein is to supply amino acids for the synthesis of proteins and other nitrogen compounds. There is no storage form of amino acids, as is the case for carbohydrates in the form of glycogen and lipids in the form of triacylglycerols. The excess intake of amino acids are metabolized, oxidized to provide energy, converted to glucose or fat. The amino acid groups are lost as urea and excreted. The protein consumption in developed countries is much higher than the recommended dietary allowance, that is 1 gram per kg body weight per day. The daily protein intake by an adult in most countries is 40 to 100 grams. Protein is digested by proteolytic enzymes to amino acids, which are absorbed in the intestine and enter the body pool of amino acids. 10 out of the 20 naturally occurring amino acids can be synthesized by the body, which contribute to the amino acid pool as non-essential amino acids. Most of the body proteins are synthesized from the amino acid pool. These include enzymes, hormones, immunoproteins, contractile proteins, etc. Many important nitrogenous compounds are produced from amino acids like porphyrins, purines, pyrimidines. About 30 gram of protein is daily utilized for this purpose. Generally, 10 to 15% of body energy requirements are met from the amino acids. The amino acids are converted to carbohydrates and fats. This becomes predominant when the protein consumption is in excess of the body requirements. Next, we are going to discuss about the metabolism of amino acids. The amino acids undergo certain common reactions like transamination followed by deamination for the liberation of ammonia. The amino group of the amino acids is utilized for the formation of urea, which is an excretory end product of protein metabolism. The carbon skeleton of the amino acid is first converted to keto acid, which meet one or more of the following fates. The fates are utilized to generate energy. The carbon skeletons are used for the synthesis of glucose. They can be diverted for the formation of fat or ketone bodies, and they may be involved in the production of non-essential amino acids. Let me explain the process of transamination in detail. The transfer of an amino group from an amino acid to a keto acid is known as transamination. This process involves the interconversion of a pair of amino acids and a pair of keto acids catalyzed by a group of enzymes called transaminases, recently called as aminotransferases. Let me explain the salient features of transamination. All transaminases require pyridoxal phosphate, a coenzyme derived from vitamin B6. Specific transaminases exist for each pair of amino and keto acids. However, only two, namely aspartate transaminase and alanine transaminase, make a significant contribution for transamination. There is no free ammonia liberated, only the transfer of amino group occurs, and transamination is reversible in nature. Transamination is very important for the redistribution of amino groups and production of non-essential amino acids as per the requirement of the cell. It involves both catabolism and anabolism of amino acids. Transamination diverts the excess amino acids towards energy generation. The amino acids undergo transamination to finally concentrate nitrogen in glutamate. Glutamate is the only amino acid that undergoes oxidative deamination to a significant extent to liberate free ammonia for urea synthesis. All amino acids except lysine, threonine, proline and hydroxyproline participate in transamination. Transamination is not restricted to alpha amino groups only. Delta amino group of ornithine can also be transaminated. Serum transaminases are important for diagnostic and prognostic purposes. What is the mechanism of transamination? Let us see. Transamination occurs in two stages. Transfer of the amino group to the coenzyme pyridoxal phosphate to form pyridoxamine phosphate. The amino group of pyridoxamine phosphate is then transferred to a keto acid to produce a new amino acid, and the enzyme with PLP, that is pyridoxal phosphate, is regenerated. All the transaminases require pyridoxal phosphate, a derivative of vitamin B6. The aldehyde group of PLP is linked with epsilon amino group of lysine residue at the active site of the enzyme, forming a Schiff's base. When an amino acid comes in contact with the enzyme, it displaces lysine and a new Schiff's base linkage is formed. The amino acid PLP Schiff base tightly binds with the enzyme by non-covalent forces. Snell and Brostein proposed a ping-pong bye-bye mechanism involving a series of intermediates in transamination reaction. Let me explain the clinical significance of transaminases. Aspartate transaminase, otherwise called as SGOT, it is the old nomenclature. The normal serum activity of this enzyme is 4 to 17 IU per liter. Concentration of the enzyme is very high in myocardium and also in liver cells. The enzyme is also distributed in other tissues, that is muscles, pancreas, kidney, etc. The enzyme is cytoplasmic and also mitochondrial. This is the enzyme which is acting as a marker enzyme to detect cardiac diseases. It is very helpful in finding out acute myocardial infarction. Alanine transaminase, otherwise called as SGPT. SGPT is the old nomenclature. The enzyme is found mainly in liver. Liver cells are rich in ALT. It is entirely cytoplasmic. The normal enzyme activity is 3 to 15 IU per liter. This is the most important marker enzyme for detecting liver diseases. Increases in both transaminases are common finding in hepatic diseases, but always SGPT is greater than SGOT. The next topic is deamination. The removal of amino group from the amino acid as ammonia is called deamination. Transamination involves only the shuffling of amino groups among amino acids. On the other hand, deamination results in the liberation of ammonia for urea synthesis. Simultaneously, the carbon skeleton of amino acids is converted to keto acids. Deamination may be either oxidative or non-oxidative. Although transamination and deamination are separately studied, they occur simultaneously, often involving glutamate as the central molecule. For this reason, some authors use the term trans-deamination while describing the reactions of transamination and deamination, particularly involving glutamate. What is oxidative deamination? Oxidative deamination is the liberation of free ammonia from the amino group of amino acids coupled with oxidation. This takes place mostly in liver and kidney. The purpose of oxidative deamination is to provide ammonia for urea synthesis and alpha keto acids for a variety of reactions including energy generation. Let me explain the role of glutamate dehydrogenase. In the process of transamination, the amino groups of most amino acids are transferred to alpha ketoglutarate to produce glutamate. Thus, glutamate serves as a collection center for amino groups in the biological system. Glutamate rapidly undergoes oxidative deamination catalyzed by glutamate dehydrogenase to liberate ammonia. This enzyme is unique in that it can utilize either NAD or NADP as a coenzyme. Conversion of glutamate to alpha ketoglutarate occurs through the formation of an intermediate alpha aminoglutarate. Glutamate dehydrogenase catalyzed reaction is important as it reversibly links up glutamate metabolism with TCA cycle through alpha ketoglutarate. Glutamate dehydrogenase is involved in both catabolic and anabolic reactions. How glutamate dehydrogenase activity is regulated? Glutamate dehydrogenase is a zinc-containing mitochondrial enzyme. It is a complex enzyme consisting of 6 identical units with a molecular weight of 56,000 each. Glutamate dehydrogenase is controlled by allosteric regulation. GTP and ATP inhibit whereas GDP and ADP activate glutamate dehydrogenase. Steroid and thyroid hormones inhibit glutamate dehydrogenase. After ingestion of a protein-rich meal, liver glutamate level is elevated. It is converted to alpha ketoglutarate with liberation of ammonia. Further, when the cellular energy levels are low, the degradation of glutamate is increased to provide alpha ketoglutarate, which enters TCA cycle to liberate energy. What is oxidative deamination? This oxidative deamination is catalyzed by amino acid oxidases. L amino acid oxidase and D amino acid oxidase are flavoproteins possessing FMN and FAD respectively. They act on the corresponding amino acids to produce alpha keto acids and ammonia. In this reaction, oxygen is reduced to hydrogen peroxide, which is later decomposed by catalase. The activity of L amino acid oxidase is much low while that of D amino acid oxidase is high in tissues, mostly liver and kidney. L amino acid oxidase does not act on glycine and dicarboxylic acids. This enzyme, due to its very low activity, does not appear to play any significant role in amino acid metabolism. The next topic is non-oxidative deamination. Some of the amino acids can be deaminated to liberate ammonia without undergoing oxidation. Amino acid dehydrases participate in non-oxidative deamination. Serine, threonine and homocysteine are the hydroxy amino acids. They undergo non-oxidative deamination catalyzed by PLP-dependent dehydrases. Amino acid desulfhydrases also participate in non-oxidative deamination. The sulfur amino acids, namely cysteine and homocysteine, undergo deamination coupled with desulfhydration to give keto acids. Deamination of histidine also occurs via non-oxidative deamination method. The enzyme histidase acts on histidine to liberate ammonia by a non-oxidative deamination process.